I hope everybody is enjoying the unit on evolution. Today we're gonna continue the journey through evolution, and we're gonna try to talk a little bit about the origin of species, how new species arise. We know that throughout time, based on the fossil record, the diversity of species has increased constantly through time. There are periods where diversity has come down. Those are the major extinction periods. But after that, we have had a rebound, and new species keep on originating. And even today, in real time, we are seeing some budding species. We can see the process actually happening. And that's what we're going to try to talk about today. So if you recall from before, we addressed the issue of populations, and if we have a particular population of individuals, and we said if you allowed enough time to pass by, because of the different pressures and the different circumstances that they are under, any of these can cause change in the populations. We can have mutations, the ultimate source of change. We can have migration as a source of change. Individuals coming to the population bringing new genes. If we have non-random matings, remember this is sexual selection basically, we are going to have a certain set of genes that are going to be preferred over that population, and those genes are the ones that are going to be passed on to the next generations. Same thing if we have genetic drift. Remember that these are chance events that happen just because of randomness. And randomness usually happens when you have, or is more significant, when you have small sample sizes. So genetic drift is usually going to be important when you have small populations, or when you have events that involve small populations, like for example, the founder effect, when a small group of individuals colonizes a new habitat, or when you have a bottleneck effect due to, again, unforeseen circumstances that nobody can survive no matter what the genes they have. So drift, pure randomness, and also we can have change in the makeup of future populations because of natural selection. Remember that of all the individuals that are present in a given population, all this set of individuals, not all of them are going to have an equal chance of reproducing. So only a small set of them, the ones with the best traits, are going to be able to survive, reproduce, and only those genes are the ones that are going to be passed on to the next generation. If this sampling of genes keeps on going and going, you can clearly see that the genetic makeup, the gene pool in this population, is clearly different from this one. Many genes here are probably not going to be passed on, and the frequencies of the different genes is going to change over time. Now we can see this in real time from generation to generation. In the lab, the favorite creatures to study these changes are, of course, bacteria with a very short reproductive time, 20 minutes to an hour, fruit flies with generation times of about 2 weeks, and even mice that have generation times of 3 to 4 weeks. So these are great organisms that allow us to see these changes in the lab and in nature in real time. And they are happening, so we can document these things. So when any of this is happening, we can see change. Now imagine what's happening as these changes start accumulating generation through generation. And remember that here we're not talking just human time frame of hundreds of years. We're talking geologic times of millions of years of these changes constantly occurring. Eventually, populations are going to become so different that at some point they are going to become reproductively isolated. One population from another, even though if they were considered the same species, at some point they're not going to be able to recognize each other because of the drastic changes in their genetic makeup. So that brings us to how the new species arise, basically through the accumulation in principle of these small changes over time. Now, to define a species, we need to start someplace, and a species is basically a group of individuals that live together in a population. Basically, a population, let's start there, is a population whose members can interbreed, can reproduce with each other, and the most important part, that they can produce viable, that means that they can survive, viable, fertile offspring. And the offspring, that's important, has to be fertile and keep on reproducing. So a group of individuals that can mate among each other, that can interbreed, and that can produce fertile offspring. That's what defines really a species. So, for example, humans, we are very different. We might look very different, all sorts of size, shapes, colors, whatever, but we can all reproduce with each other, and we can all produce fertile offspring regardless of our looks that are heavily influenced by the environment, especially skin color, as I hope you have read in the article about skin color. Here you have two birds, the eastern meadowlark and the western meadowlark. You look at them, they look exactly the same, very similar. Yeah, they look the same, but actually they cannot reproduce because they cannot recognize their mating rituals. So even though they look the same, they are really, truly different species because they cannot reproduce with each other. So looks are very deceiving, so looks are not the important thing that is going to allow us to recognize species. Is the reproductive potential and producing viable offspring. And that definition, that idea of species was proposed by Ernst Mayr, one of the pioneers in evolutionary studies. Right. Now, when we talk about speciation, basically speciation is nothing else but the origin of new species, how new species originate. Sometimes they divide speciation into two different types of events, something that they call microevolution, small level evolution, and that's kind of the changes that we see in populations, small little changes in the genetic makeup of populations. These are very basically at the gene pool level, and they are very small changes from time to time. Macroevolution, the big evolution, they usually refer to this when there are significant changes in the morphology, and basically that allows you to clearly identify changes at the species level and above the species level. You recall genus, families, orders, even phylum. So those major changes that produce significant changes, those are usually referred as macroevolution. But in general, macroevolution is a product, basically, of the accumulation of all these small changes over millions of years. You change enough that from time to time, it's like when your grandparents come to see you and they have not seen you in 10 years, they go, "Oh my God, how have you changed?" The same process. Your parents that see you every day, they don't see the major difference. They just see little changes day to day. But distant relatives, I see you every 10, 20 years, they go, "Oh my God." That is kind of a good analogy for macroevolution. Now, for speciation, how a species change into another, there are two basic patterns that we can quickly mention, just if you come across these names: anagenesis and cladogenesis. It's not critical that you remember this, but it's always useful because it gives you a big picture of how things happen in time. If you look at the fossil record, sometimes you see a species, and that species, of course, through the fossil record changes very slowly from one into other because the environmental conditions are changing, but you don't see any other similar species. It's just one kind of slowly morphing into another one through time. That is called anagenesis. And basically, it does not increase the diversity of species because you have one species here and you still have one species different later in time. It's just a kind of a terminology and semantic thing, but it's good to understand this. So anagenesis, the change over time from one into another, does not lead to diversity. However, the most common pattern that we see in the fossil record and that we can see every day is what is called cladogenesis. Cladogenesis. And this is the speciation event in which the gene pool of a particular species eventually splits, and this species actually becomes two different species over time. This pattern that we see here, this is the one that increases diversity of species and increases the number of species over time. And this seems to be the most common pattern of speciation that we're going to see. So don't worry too much if you don't truly get anagenesis and cladogenesis. You're going to get it as we continue in this unit and you become more familiar with fossil records and the history of some species. So, how and where and why new species originate? The main process, remember that species, you have to start basically with a population, a group of organisms that can interbreed, and usually in order to interbreed you kind of need to live together. Now the main requirement for the new species to arise is that at some point the populations are going to become isolated. Populations are going to separate, but does not have to be geographically necessarily. Can be anything, but the most important thing is that reproductively they are isolated. They cannot interbreed. And the important thing is that if they cannot interbreed, their gene pools are not going to mix. So they are not going to spread around evenly. Their gene pools are going to become two different gene pools that are going to follow different paths. And that's the important thing. The separation of the gene pools that is going to make each one of them unique because of the different conditions that they are going to face. So, reproductive isolation is the key element for the origin of new species, because that is going to separate the gene pool. This is going to allow the species to evolve independently, to change. Remember, evolve means to change independently. And because of that, actually, we have the two main types of speciation that we see these days for real in nature. One type of speciation that we are going to call allopatric speciation, that this involves some type of geographical, physical isolation of the two populations to become two different species. And then we're going to have sympatric speciation, where actually all the individuals are still living in the same place, but somehow within that same place there is like a clique, if you wish, of individuals that don't like to mingle with the rest of the individuals in that population. So truly physical separation for allopatric speciation, like here, truly physical separation of the original population. And in sympatric speciation, they are still together, but somehow there is a small clique that develops, and though that small group of individuals, for whatever reason, they just reproduce among themselves and have nothing to do with the other ones. So again, the critical concept of being reproductively isolated. If you are reproductively isolated, you cannot mix your genes, and that is going to lead to two separate species over time. So you have this nice diagram in your textbook. I also gave you a little diagram with this. I recommend that you take a minute to jot down a list of these. These are mechanisms that enforce reproductive isolation. Or these are things that are going to cause some type of reproductive isolation. So this list that we are going to go through, these barriers to reproduction, they can apply at the very beginning, things that are going to cause this separation, but they can also be used after the separation has occurred to reinforce this separation. So you can see them as a pre or a post kind of thing to speciation. You can use them anytime. So we are going to have some of these are what we call prezygotic barriers. So stop, take a minute, copy this down, and identify the prezygotic and the postzygotic barriers, so you can have a nice outline and you can keep your facts straight in your head. So prezygotic barriers, these are things that are going to happen that prevent the mixing of the genes. It's a nice reproductive barrier that happens before the formation of the zygote. So everything up to the development of the zygote. And the goal of these prezygotic barriers is basically to save you energy and time. If the zygote has no future, what's the point on wasting energy for this species? It's not an evolutionary smart process, so those that waste their time have been eliminated. So again, prezygotic barriers, barriers that prevent the development of the zygote because you are going to become reproductively isolated. And postzygotic barriers, okay, we had fertilization, somehow none of these barriers work. Even postzygotic, there are components that are going to keep these species separate and prevent them from becoming one again. So let's go one by one. So we have, in the prezygotic barriers, we have geographic barriers and habitat isolation, temporal isolation, behavioral, mechanical, and gametic isolation. Gametic isolation. So we'll go through all of these. If you are very good at this, you might just want to read about it. Otherwise, keep on watching. What I'm going to try to do is give you a couple of examples of each one so they can stick to your head. But reading is still the best option here. So examples of geographic or habitat isolation, basic barriers that separate two populations and that maintain them separated in such a way that they cannot reproduce anymore. They cannot mix their genes. And if they cannot mix their genes, that means that over time they're going to start looking more and more different. The first example that we have here is the squirrels that live in the Grand Canyon area. As you might know, the Grand Canyon is a high plateau of sedimentary rock, and the Grand Canyon is about several million years old, 6, 7, 8 million years old. I don't recall the exact number, but basically that many million years ago, 5 or 6 million years ago, the Colorado River was right here at the very surface. And through the last 5, 6, 7 million years, basically the Colorado River has been digging a hole and is still digging a hole deeper, basically forming the canyon. So the Grand Canyon is getting deeper anyway, and it's going to continue getting deeper. So at the beginning, when the Colorado River was just a river on the surface, small river, there was one species of squirrel that could jump around just like our squirrels around here, and one original species that occupied all that area. In the last 5, 6 million years, as the canyon got deeper, the movement of squirrels got limited, and with that, of course, their reproductive capabilities. So what do we see right now, present time? There are two different species, actually, of squirrels that live on opposite sides of the Grand Canyon, the North Rim and the South Rim. They still look very similar, but their mating behaviors and are totally different. So even if you kind of put them together, they kind of don't know what to do with each other. So effectively, reproductively isolated because of a nice geographic barrier in the Grand Canyon. We can also have this geographical isolation due to ecological factors, the type of habitat that they live. As you know, lions and tigers, you find them both in Africa. However, and they're close relatives, they both belong to the same genus, Felis. Now, what's the difference between these two? One of the differences is that they occupy, even if in nature, in real nature, they have very few chances of coming together. Because in nature, lions live in the open savannas and tigers live in the forested areas. So in reality, they are reproductively isolated because of the type of habitats that they occupy. Even though we know that if we put them together in a zoo where they lose all their instincts, and not all their instincts, but their habitat is not the same, they cannot do their own thing. You know, being enclosed in a little cage does funny things to everybody. And we know that in captivity we can make these two kind of mate. But in nature doesn't happen because they actually live, they are isolated because they live in two very different habitats. So two different species, and the longer they remain in these two different habitats, the more and more differences are going to arise that are going to keep on making the species more and more genetically different. Have another case of same kind of topic, habitat issue. There are two species of garter snakes, genus Tamophis. They live in the same area, so altogether if you find one species in the ponds, in the water, one species that prefers to live in the solid ground, and they live together in the same area, but one prefers to do all its business in the water, the other one prefers to do all its business mostly on land. So again, they might live in the same area, but effectively they are kind of isolated because of the different habitat preferences that they have. Now another factor here that increases or facilitates reproductive isolation and the formation of new species is what we call temporal isolation. For most mammals there is some type of reproductive behavior, courtship and stuff like that. Well, in a lot of animals, the breeding season between closely related species is different. Early in the season, later in the season, and that difference is gonna separate the gene pools of organisms. We have the example of the eastern spotted skunk versus the western spotted skunk. They look very similar because at one point they were truly different species, truly one same species. But one reproduces in the late winter and the other one reproduces in the late summer. So when one is ready to reproduce, the other one is no near close. So again, those kind of barriers enforce the separation of the species and keep them separate. This is reproductive isolation, but I want to introduce you again to a possible example of sympatric speciation. Sympatric, remember sympatric is when the individuals occupy the same habitat, they live in the same area, but for some reason they start separating the gene pools. And this seems to have been an example where the ranges, the eastern and the western skunk overlap. There is that separation in mating times that is reinforcing the separation of the species. Let me go back a second, because another typical case of temporal isolation with reproduction has to do with plants. Many plants have very specific times times of the day at which they bloom, where the flowers open up. There are very similar species sometimes in which one species opens up early in the morning and is available for pollination early in the morning, and as the day progresses those flowers close and another very closely related species opens up in the evening. So effectively there is no chance for these two species to mix up, and that separation in the time of blooming, basically in the day, enforces the separation, the barrier that causes reproductive isolation, and enforces and can also could have been a reason for reproductive isolation, just the separation in blooming times between two different populations. So I want you to realize that this goes both ways. You can start with a population that blooms at different times, even though they live in the same area. That's going to cause these two populations to diverge because they are not mixing their genes. Now, once their separation arises, this separation in time of blooming is going to maintain those two species as separate over time, increasing their genetic separation. Behavioral isolation is a very strong one, especially in organisms that have sexual selection. because the mating patterns are so defined and so specific and so species specific that if you don't do the right thing that your species is expecting, you're not going to get anywhere. So here is any of the species that you know that have sexual selection falls into this category. But as a caveat, this is also an important factor for sympatric speciation. Just imagine for a moment, and this is a totally hypothetical example, but that can help. Imagine for a moment that you have a nice pond here with frogs. You have tons of male frogs. All these frogs are croaking, singing their song, and there is one population. Now imagine that given to a mutation in the area of the vocal, they don't have vocal cords, but the sound producing organ in frogs, due to a particular mutation, this particular male out here produces a very different croaking sound. If you are a female, most females, of course, are going to flock to the typical one. But if you have a female that is different, maybe that doesn't have very good hearing after all. You know, there is variability in a population from all over the place. You have a female that doesn't have very good hearing, cannot very well discern or notice the differences. If that one odd female mates with this male that has a totally different courtship song, guess what's going to happen here? If we have that case that two individuals act very differently from the rest of the population, guess what their offspring are going to inherit? Their offspring are going to inherit the bad or odd genes for the son, for the father, and their offspring are going to inherit probably the poor sound capabilities, discerning capabilities of the mother. So right there in the next generation you have a pool of individuals. Remember, they produce many offspring. You have a pool of individuals that is slightly different than the other ones. And you know, this could be the beginning of a new species, a new clique, if you wish, because they are not going to fit necessarily in the pattern of the other ones from the point of searching for mates. Obviously, they are going to like frogs that are more like themselves. And again, since we know that there is extreme variability in a population, there's probably going to be some odd ones out that are going to fit into this pattern. And right there, you have one species and maybe a new species due to these small differences. Since all this is happening in one area, that's a typical example of sympatric speciation. And this, I'm giving you just a very vague hypothetical example, but this has been shown to happen in nature over and over and over again. I have a book here called Speciation and its Consequences, and basically the whole book is just one example after another of different speciation events that have been documented and how they have happened. So I talk to you in very hypothetical terms, but there is evidence of all this. Now, the courtship behaviors and displays are not only for birds. So the courtship behavior, a change in the courtship can be the beginning of a new species, but it can also mean, excuse me, it can also mean that it's a mechanism to reinforce the unity of our species. We're very familiar with the big mammals, but I might want to introduce you to two other ones. Lacewings are very small insects that obviously they all look the same to us. All the different species look exactly the same morphologically, but their courtship song, the sound that they produce, kind of like the cicadas, is very species specific. Here you have the sonograms, the frequencies of the different calls for 3 different species. Notice it's the same genus, different species, and you can see the differences. So this species is never gonna mate with that one. So again, many mechanisms to reinforce that this separation or reproductive isolation. If you are familiar with fireflies, you know that during the summer month you can see them with their lightning lights in the abdomen. Notice, for the record, the females usually stayed in the ground, and the males are the ones that are flying out there showing off their lights. The females are sitting quietly. When the females see a male that catches their attention because they like the intensity of the light or whatever, then the female lights up and said, "Hey, I'm here." And the male, zoom, and they copulate. So now, each species, we have several species in our area. We have about 7 different species, and the 7 different species have different types of frequencies. There is one that goes light, light, light at about 3 or 4 second intervals. That seems to be the most common. There is another one that lives more near bushes and goes light, 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 light. The pattern and the intensity, the color of the light is very distinctive for each one of the species. So if you want to know more about it, Google it or come talk to me, but it's a fascinating topic about fireflies. So again, males do their signaling on top, females are on the ground. When the females see a male that catches their attention, the females light up, the male recognizes the female, sees it, and mating occurs. So, geographical, temporal, all sorts of things. Now we have one that is very, very interesting, and is mechanical isolation. Mechanical isolation means that the parts, remember, all these are prezygotic, all these things that try to prevent 2 species from mating, that start separating these species, or once they become 2 species, kind of maintains their separation. So the next one is mechanical isolation, and this has to do actually with the morphology of their sexual organs, the mechanics of it. Plants, you know very well that at least plants that are pollinated by insects, the morphology of the plant, the position of the pollen, and the position of the stigma, the sticky stigma, the female part that receives the pollen, have very specific morphologies, and they are all adapted, basically very well adapted to the type of pollinators. So if you are a hummingbird, the flowers offer nectar that is at the very end of the flower, so that the bird has to go in and get their face all full of pollen before it moves to the next one. So it can get the food, but you are getting pollen all over. That's a trade-off. Same thing with bees. They are all very, very specific. Be they birds, bees, flies, beetles, anything that is used for pollination, they are very, very species specific. Now, the morphology changes. So if you happen to have a mutation that changes your morphology, it's going to change your pollinators. By the same token, specific morphologies are basically for specific pollinators that feed on specific plants, ensuring the transfer of pollen from specific plants to another. So this specificity between pollinators and flowers is another one that reinforces this mechanical reproductive isolation. So plants are okay, but the most interesting ones are obviously the animals when they have mechanical isolation. The penis and or the equivalent of the penis and the equivalent of the female opening have to be a match for fertilization to occur. And I don't know if you're aware of, but in many insects, identifying the species is done, they look the same from the outside, The identification of the different species is done by the morphology of the reproductive organs. In moths, if you want to identify many different species of moth that are closely related, you have to go and make a squash preparation basically of the reproductive organs to be able to identify them. It's fascinating. So here, for example, you have the damselflies. We are all familiar with the damselflies. The penises are very different between different species. And one type of penis only fits into the correct female. So again, mechanical isolation. In case there is confusion, the mechanics don't fail. So, the habitat didn't stop you for trying to mate, the behavior didn't stop you, the mechanics didn't stop you. There is always what is called the next step. So all these are various steps, from the most obvious, just living in different habitats, to the most, okay, we are getting closer to intimacy here. The next one, you are getting closer to fertilization. This is gametic isolation. Basically, this allows the gametes or the eggs of one species to be able to be fertilized only by the sperm of the same species. There is, remember, as any other cell, they have receptors in their surface and the sperm has markers to receptors that fit only with the specific type of receptor in the egg surface. So this is particularly true for species like sea urchins, starfish, many worms, corals that just release their gametes into the water in the ocean. Just imagine that soup of ovules and eggs and sperm swimming around. Major confusion. But it's not such, because the markers on the surface of the egg and the specific receptors in the sperm are such that allow for perfect matching, perfect cell communication there. So if you want to generalize, we can have biochemical barriers where the sperm cannot penetrate an egg that is not the right one, because there is no good receptor recognition. The molecules on the sperm and the egg do not recognize each other. And besides that, there is also some chemical incompatibility. The sperm sometimes cannot survive in the reproductive tract. The biochemistry of the chemistry of the reproductive tract is such that has a specific pH, specific conditions that allow the sperm of only specific organisms to survive. And even those are always in danger, because white blood cells and other things are always trying to kill these foreign organisms, quotations, that are in the reproductive system. So again, even though everything else fails, you are trying to have fertilization, there are these biochemical barriers that impede the production of, or the waste of, your valuable reproductive resources. Now, these are things that emphasize the separation of the species. Now, what happens if all that fails? All these barriers that we have by nature that are in place to prevent the mixing or to encourage the separation because of differences. What happens if all those pre-reproductive barriers, prezygotic barriers, fail? Well, you can have fertilization, but even after fertilization occurs, there are still mechanisms to say, no, you're not going anywhere. So, for example, we have three main postzygotic barriers, and one of them you are very familiar with. These are all barriers that are going to prevent the hybrids. Hybrid is usually the name given to an organism that is the product of two different parents. In this context of speciation, is an organism that is the product of two different species. So has the genes of two different species. Now, the first level of stopping the system here is what we call reduced hybrid viability. And what happens in reduced hybrid viability, if you have two different species mating, and maybe they have just become different in the recent past, so they are not totally completely isolated. So the barriers have not been enforced that much. So they actually mate, they have an offspring, but their offspring, the hybrids that they produce, have reduced viability. So basically they are not going to live very well. And that's, for example, we have many species of salamanders that live in the rivers that are very closely related because they just started the process of speciation not too long ago, and because rivers change course and the topography changes with formation of mountains and other things, sometimes rivers that are isolated eventually become reunited again. If this isolation was not long enough to truly separate two different populations and these populations reunite again, sometimes what happens is they will mate. They are close enough that they will still mate and they will produce offspring, but the offspring has very low viability and most of them die early on during development. So yeah, you have a baby, but the baby dies. We're going to put it in very simple terms. You have a baby and the baby dies. That's reduced hybrid viability. So by eliminating all those hybrids, basically you maintain the pure species genome. The second step, you mate, you produce a baby, and the baby doesn't die. The baby survives. And the baby survives and is a mule, for example. Gets to live a happy life. However, what's the problem? The mule cannot reproduce, is sterile. So, yes, you get to live longer, but at the end of the day, it's the same process. You are sterile and your genes are going nowhere. So this is reduced hybrid fertility. First one was viability. You die early on. Fertility, you get to live, but you cannot reproduce. And the reason why the mule actually is infertile is very similar to the situation that I explained between what happened between monkeys, primates in general, and human chromosomes. We have 46. Most of our close primates have 48. Well, horses have 64 chromosomes, or 32 pairs. Donkeys have 62, or 31 pairs. So when the gametes join, you have 32 in one of the gametes, 31 in the other one, and basically you're always going to have one unmatched. And that's what prevents this organism from being fertile. In most cases, a situation like this in your chromosomes, the whole organism doesn't make it. So it's kind of amazing that you still get a mule out of this. So what's the third level? So first, the babies die. Second level, the babies get to grow, but they never get to reproduce, so it's a dead end anyway. The third level of postzygotic barrier is the babies survive, the babies get to reproduce, but the baby's offspring doesn't make it. So you almost make it, you have your babies, and that's the end of the road. So you're allowed to go one further generation, and that's the end. And that's in here. These are hybrids, and whenever they reproduce, these are the babies in plants. We can see this mostly in plants. We rarely see this level going to the next generation in animals. And of course, the next generation, this is totally inviable and pretty worthless. Doesn't go anywhere. Okay? So the three levels, and we have seen in rice, this is a specific example. If you have two hybrids trying to reproduce, the next generation is small and sterile. So that's the end of the road. So all these have been a series of barriers, and again, I want to think you about this in evolutionary terms. The meaning of it is all about trying to maintain the separation of species because of reproductive isolation. And this reproductive isolation can be viewed from 2 points of view. Can be viewed as the reason that these two species are forming, are separating, or after these species have been separated, as the reason to prevent them from joining back together again. So we have speciation. So let's review now, because I mentioned a couple of times this allopatric. In allopatric speciation, again, this is very important and you have to conceptualize this, is basically the separation, the physical separation of species because of some type of geographic barrier. The Rocky Mountains were a great geographic barrier here in America, separated the creatures from the east and the west. When the Central America, the isthmus of Central America emerged, that has been in the last 20 million years, that actually separated an ocean. The Caribbean or the Atlantic and Pacific were one ocean altogether until through volcanic activity, the Central America came up. And when Central America came up, all the organisms in those two areas separated. Now guess what? The closest relatives of the organisms in the Caribbean are found in the Pacific coast of Central America. As this, we have many other barriers. Rivers can be enormous barriers for small organisms. It's no big deal for a big creature, but for a small organism, a river can be enough to isolate populations. Now, the second type of speciation that I mentioned is sympatric, and this is speciation that occurs without truly isolating two populations. The organisms remain in one place, but for some reason a small group of them becomes isolated, reproductively isolated from the other one. And reasons for this could be small mutations that cause changes in the courtship behavior, mutations that cause changes in the timing of their reproduction between either daily, morning versus night, or time of the year. And those can be triggered by just simple mutations. Now, besides this non-random mating that can change things around, there is another change that can cause sympatric speciation. And these are chromosomal changes, very fast changes in your chromosome makeup that basically almost instantaneously renders the two groups of individuals totally different. And these chromosomal changes are usually associated with plants and are called polyploidy. Remember, most organisms are diploid. We get one set of chromosomes from mom, one set of chromosomes from dad. So we are diploid. Our gametes are haploid. Polyploid means that you have multiple sets of the same chromosomes. How do you get there? Well, there are several mechanisms, but one of the most common ones is that when here we have an organism with 6 chromosomes, 2 and 6, or just 3 pairs, you know that before meiosis, before the formation of gametes, everything duplicates during the S phase of cell division. They duplicate, and then these are supposed to go through 2 divisions of meiosis until they separate. Well, if there are problems with the mitotic spindle, they might not separate. They might not separate. And you might end up with gametes, or totally, you might have meiosis 1, but not meiosis 2, for example. If you have that, you are going to end up with gametes that have double the number of chromosomes. So imagine this is S phase, and you just go through the first division of meiosis when homologous separate. You're going to end up with doubled up chromosomes, with 6 chromosomes here, 3 pairs. Now, if these 3 pairs are your gametes now, because they are not going to go through the second division of meiosis, and if you are able to find another one like this near you and you fertilize it, what are you going to have? Immediately, you are going to have an organism that is 4N. You go from 2N gametes because of a problem during meiosis. When 2N gametes fertilize each other, you get 4N. Basically, you get offspring that is tetraploid. When these offspring make gametes, of course, those gametes will never work again with a normal individual of this population. So technically, instantaneously, you have right there a new species. Where do we see this type of phenomenon happening? For some reason, plants love to do, no, they don't love to do this, but this happens in plants often, very, very often. And a lot of plant species have arisen basically because of problems during their meiosis, and they are polyploids. For some reasons, plants are not very good at recognizing good and bad mixes here, and they do make a lot of mistakes, and they do fertilize a lot the wrong things. So in this case, you have a tetraploid right there. It's a new species because of the reproductive isolation trying to mate with the original population. Now, if you recall, many plants, most plants are hermaphrodites. So they produce male and female gametes. Now, even though most of them are hermaphrodites, actually most of them don't like or are incapable of fertilizing themselves. They like cross-fertilization because that's how they increase their genetic diversity. However, there is always a few of them that will do self-fertilization, and they will fertilize themselves. So if you think of a plant that has just gone through this problem in meiosis, is totally screwed up, is fertilizing itself, hey, both eggs and sperm might be defective. Perfect. So you don't have to go anywhere. Right there, through self-fertilization, you have bad eggs with bad sperm, or 2N eggs and 2N sperm fertilizing each other, and right there you have a new species just by yourself. Now, if this plant that is tetraploid now continues doing the self-fertilization, let's assume now that meiosis is going to occur normally. From now on, you have a whole new population, a whole new species. This that I just told you was documented in Washington State about 70 years ago. They were studying, I believe, some grasses, and all of a sudden they find this new one that they had not seen. They start monitoring the grass, they do all the genetic analysis, and they realize that it's just a polyploid of the grasses similar to the grasses that were around, but they were not mating. So in the spot, bingo, new species. So polyploidy, very, very common in plants. And polyploidy, if you want to dwell, I put something in your math notes. There are 2 types of polyploidy. Autopolyploidy, auto, yourself. This is kind of the one that I described. And there is also autopolyploidy, the one that we have here. And there is also allopolyploidy, allo meaning with others, that instead of just doing it with yourself, basically you are finding actually other individuals with whom to fertilize your egg and sperm. And a common example for that, actually, very popular, is what we call wheat, our bread wheat. Bread wheat, now massive production of it, but if you try to follow where wheat comes from, this doesn't really exist in nature. This has been developed in the last 10,000–12,000 years, and they have tried to figure out how it came to be. And in reality, there are 3 different species: Triticum monococcum, another type of wild Triticum, Triticum is the genus, and another one. So there are 3 species of wheat actually or wheat-like plants, with all of them with 14 chromosomes, 2n14, haploid number 7 pairs. And what they have realized, so this is a diploid set. This is a diploid set for this species, and this is a diploid set for this species. Right? So by studying what has happened to the wheat chromosomes, when you look at our modern day wheat, you are going to realize that our wheat has the genomes of 3 different species altogether. Species, this one, all the chromosomes, plus all the chromosomes from this, plus all the chromosomes from this. How many chromosomes is that? 14, 28, 42. Our modern day wheat really is the product of joining the DNA of 3 different species of wild wheat. And that is Triticum stivum, what we call the bread wheat, the one that is high in gluten, that is not good these days. Good? Excellent. So this is all polyploidy, very common in plants, very uncommon in animals, but there are a few animals that do it. Why that is, not very clear. We are just in the process of figuring all these things out. So plenty of research ahead of us. So that brings us to kind of the end here. So we have looked at things that favor the reproductive separation of species, and after the species kind of become separated and their gene pools go different ways, these same factors could be used to kind of reinforce the separation of these species, and over time they become new species. So from one, when they separate, they are going to become 2 different ones, because they are exposed to different environmental pressures. Now, how this process happens when you look at the millions of years of the fossil record. For a long time, when people were discussing, or paleontologists were discussing how species evolve, in many species they found that the process of change in the fossil record was very gradual. You could see the morphology of the fossils changing slowly through time, from 50 million years ago to the present, or even from 500 million years ago all the way to the present, a very slow process of change illustrated in this diagram. This process of change is what we call gradualism, and has to do with the rate of speciation, how fast it's happening. It's happening slow, very steady, and the assumption, remember that all these changes, most of these morphological or changes in populations are driven by the environment, because it's the adaptations and who gets to reproduce that matters. So when you see organisms changing this way very slowly, here is a common ancestor, and here we have an event that splits this population and keeps on going different ways very slowly. The assumption here is that the environment is changing slowly also through time. Like in our lives, we notice that things are changing slowly, and that's gradualism. So if you have here, you have time and morphological change. From the beginning, very gradual morphological change as time goes by, because the environment is assumed to change slowly. Right? So in this case, any big changes, if you compare the top ones to the original ones, yes, you're going to see a big change. But in between, everything was gradual. Good? Now this is gradualism. And in contrast to gradualism, there is something called punctuated equilibrium. I love that, punctuated equilibrium. If you pronounce it right, it sounds perfect. This was proposed actually by a paleontologist at Harvard that unfortunately died not too long ago, Stephen Jay Gould. He was so good that he even made it to The Simpsons. There is a whole Simpsons episode with Stephen Jay Gould. You should Google it and watch it. So he studied fossils too, he was at Harvard, and he noticed actually something different from this gradual. He would study a species, and for millions of years this species was exactly the same. And then all of a sudden, in a very short period of time, just a couple of millions, everything is relative, boom! Humongous change in the morphology of the species. So instead of being a constant gradual process, what he saw in this species was same, change. Same, change. And he called this punctuated equilibrium. So same, change. Same, change. And so basically his idea is that for most part, basically, if the species remain the same, that means that in these particular environments, the environment is pretty constant all the time. And then for some reason, environmental something, volcanic eruptions, meteorites, all those upheavals that we have on Earth, basically something changed drastically and put a major pressure onto the species, major selective forces that force them to change very fast in a very small period of time. Small in geological time, just a couple million years. So make sure that you understand those two, gradualism and punctuated equilibrium, the two different ones. So rapid change, when they first change from a population, and that is because always the driving force is the environment. However, if you have polyploidy happening, is polyploidy gradualism or is polyploidy punctuated equilibrium? Think about it. Well, polyploidy will be one of these punctuated equilibriums. Same, and then all of a sudden, hey, you have a major change because a major chromosomal rearrangement had occurred. Alright, so I hope that helps you to understand this is related to the rate of evolution. The other things that we talked were related to what contributes to the separation of species. Now, the big point here now, just to finish up, is species change. Where do these new things that we see all the time happening come from? Remember, anything that we see, even something as complex as an eye, a human or a mammal eye, that is one of the most complex structures, you can actually follow its development back in time and track how small changes have been accumulating into this structure to produce what we have now as the beautiful, perfect eye. You never go from something very primitive to something extremely advanced. You can actually follow, in evolutionary terms, if you look hard enough, you can follow all these processes. And you have evidence for that in the fossil record. And when you don't have evidence, remember that the fossil record is imperfect, that we have not found all the fossils, and that not all organisms fossilize. But if we keep on looking, we'll find enough to fill in enough, as we have done so far. The other important concept here about how these massive changes happen, sometimes you realize, how can you go from a snake to a dog? I mean, those are significant morphological changes. Well, they're not closely related, I'm just exaggerating. But a lot of changes, you don't have to reinvent the wheel. You have the genes. The only thing that needs to be changed sometimes is the speed at which some structures developed in your body. So here you have, for example, this is, you don't need to remember heterochrony or anything, but has to do with the genes that regulate development. Those master regulatory genes are called homeotic genes, or HOX genes, H-O-X. Look at the cranium of a chimp and a human. We look very similar when we are fetuses. What happens through development to make our head look so different? It's just the rate at which different bones grow that gives us the different shapes in the head. And the same is true for most mammals. because we have all exactly the same bones everywhere. It's just the rate, the speed at which each one of our bones develop that gives us the different morphology. And those rates are just regulated by a few genes in this group of very important genes that we call homeotic genes, that are the master genes that control development. Perhaps the most fascinating for me, I love this. This is a nice salamander. It's called the axolot. And what it is basically, it looks like a baby salamander because it maintains all the baby-looking structures. But what's happening here is sexually mature. So the body remains baby-looking, while inside, basically all the organs mature very, very fast. Just imagine a little human baby being sexually mature. This is the axolot. Basically retains all the baby infant characters, but when you look at it inside, it's totally sexually mature, ready to reproduce. So again, it's the timing of the things, the mismatch between physiological development and body development, and that right there is a great source of morphological diversity in organisms. Good? Well, so to finalize this, please remember that evolution is just change over time, and when you start changing things, when you start tinkering with things, you can only do it with things that you have. Evolution does not say, oh, for next generation I'm gonna need a new set of arms so I can make you different. Evolution, this change, works with what you have, and that is best. And that's why, even today, humans are not very perfect, because since we became bipedal, a lot of problems come because we became bipedal. We have back problems, we have hip problems, we have walking problems, knee problems. Girls, because of the wide hips, females tend to have more issues with knee issues. Did I say that? Okay. So you work with what you have, but you're always going to have some baggage, because you cannot make anything perfect, because you are just starting and modifying what you have. Henry Ford didn't create the Ford Mustang right off the blue, or the humongous pickup truck. He started with Model T. And through 100 years, we have been improving on that Model T until we have this gorgeous Mustang or these humongous pickup trucks. But they never started that way. It's a slow process of improving over what you have. I hope that helps. Speciation is awesome, and we owe the diversity on Earth to this process of speciation.